And okay, and we're live now with our first Journal Club discussion of 2016 for our Microbiology Journal Club. And today we're going to be talking about this paper. Cpf1 is a single RNA-guided endonuclease. How am I getting? Now my audio is off. Can anybody else hear that?
I can hear you.
Good, you're there.
Okay, hang on a second. Oh, I know what's going on. I can hear myself. Haha, I had my YouTube video open and it started streaming and I could hear it through my headphones. I was like, why can I suddenly hear myself? Okay, everything has been fixed now. Technical glitch. So this is the paper we're discussing today about CRISPR and Cas systems, which I'm very much looking forward to. After our technical problem. So I am Laura Williams. I'm an assistant professor at Providence College in Providence, Rhode Island. And I am in my third semester now, so this is a very exciting time for us. We're getting our research going, and I'm starting to feel like I have a little bit of a handle on teaching, which is probably all an illusion because it's only week 2. So that'll probably collapse pretty rapidly. I'll have Mark go next because he's right next to me. So Mark, do you want to introduce yourself?
Sure. I would normally have a wonderful little logo at the bottom, you know, with sparkly lights and all the rest. I'm Mark Martin, an associate professor of biology at the University of Puget Sound in scenic Tacoma, Washington. Not snowy, however, unlike Providence, I presume. And I, as an undergraduate educator, teach Introduction to Cell and Molecular Biology, Microbiology, and Research Methods. In my small and modest research lab, we study predatory bacteria a little bit here and there, as well as bacterial symbioses and related issues.
Great, thank you very much, and we're glad to have you with us. So next to Mark is Joel, who's a bit new to our group. So Joel, do you want to unmute yourself, and then you can tell us a little bit about you?
Sure. I'm Joel Graff. I just started as an assistant professor at Montana Tech. Previous to that, I did my postdoc and then worked my way up to research associate professor at the University of Iowa. So 3 weeks of being at Montana Tech. I was 100% research before, and now I'm pretty much all teaching. They're taking it easy on me. I'm teaching freshman level cell biology, and I'm teaching an upper level immunology class. So I think I'm more infectious disease and immune system than a lot of people that are in this journal club typically. And my PhD work was studying rotavirus, and my postdoc work was studying a parasite called Leishmania, and I have a particular interest in macrophages, the big eaters in your immune system. And I am trying to get more into science communication through Twitter, which I had never touched till a couple weeks ago.
Yeah, we sucked you into Twitter.
Yeah, and then I started a YouTube channel. It's called Life in Biology, and basically I kind of got addicted to woodworking channels on YouTube, and I decided people upload a new video fairly often. And like a weekly video showing a project, and I thought, well, how about I do like a weekly video doing some science in the news story and try to break it down and make it a bit entertaining for people. So that's my foray into science communication. And then I just searched around on YouTube one day looking to see if people post journal clubs, and this looked great, so I contacted Laura and Karen and joined in.
Yeah, thanks very much for joining us.
Yeah.
And then last but not least, we've got Dave with us. Dave, do you want to say a little bit about yourself?
Dave Baltris. I'm an assistant professor at the University of Arizona, where it was 84 degrees last Saturday while everybody else was under the snow. It was good. I sat on the couch watching the Weather Channel with my popcorn. It was like watching a movie. I am currently a fifth year professor at U of A, so I go up for tenure in July.
Oh, wow.
And I teach upper level microbial genetics and 3 labs, 2 of which are running right now. So if I run out early, I'm going to check on my TAs in the lab. So yeah, so that is it.
Okay. Yeah, no problem. If you have to skip out, go ahead. You know, as I've mentioned before, this is pretty informal, so we sometimes have people who pop in late and people who have to pop out early, and that's not a problem at all. We're fine with that. So if there's anybody who's watching right now who's a little shy about joining because you're afraid of time or so forth, feel free to hop in. We're happy to have people join us for the discussion. Okay, so we'll hop right into it. This paper was suggested by Joel. Thank you for the suggestion. I think it's a really good one and a really, as it turns out, a very timely paper, because it has to do with CRISPR-Cas systems. Now, what is the preferred pronunciation for the Cas? Is it Cas? Cas?
Yeah, I say Cas.
Cas? That's how I said it, but I just wanted to be sure.
Sometimes I have it with a little bit of a Z sound.
Okay.
I don't know.
All right, we'll accept both for the purposes of this journal club, I guess. All right, so, you know, if you've been following the news, the reason that CRISPR is making headlines is, in one way, it's kind of a good thing, because it's kind of exploded the amount of research that's being done on these systems for their potential in genome editing technology. The idea here being that if you can tweak these systems that occur in bacteria and archaea, if you can tweak them for editing in mammalian cells, this could become part of a very advanced therapy for different diseases that have genetic origins.
No.
What?
That's not the reason.
That's not the reason?
So, sorry, I have my 5-second rant on this.
Well, I was going to get to the other reason, but
No. So there is no reason to use CRISPRs for genome modification in humans. Because you can screen
elaborate.
Yeah, well, you have to do everything in vitro anyway, so you can just sequence the embryos and screen for the right mutations. So the only reason to use CRISPRs in humans is to give them something that is not already there. But that is it's an agenda based on people. But nonetheless.
Okay, so I mean, so do you think that it's been kind of misrepresented in terms of just what its potential applications are, or do you think that this is?
So the people are selling it because it's an easy story to be like, oh, see, that there's this person with a horrible genetic disease that CRISPR can cure. Well, that's not really true, because if you need to get it in all of the cells of the body to actually cure it. And so, I mean, I can see something maybe with stem cells where they can re-inject them, but to actually say we're curing disease in generations going forward, you can just sequence the embryos and screen out you have to do in vitro embryo fertilization anyway, so you can just screen the ones that don't have the diseases anyway. So really, everybody is billing it for this, like, we can help humanity, but really there's going to be, it's more to impart other things versus the ones that are already in your genome.
Dave, I really want you to have a thing at the bottom that says brilliant iconoclast, because that's a really good description of you.
I just like arguing, that's all.
No, I mean, that's not entirely true. You just have very, very strong feelings, and you know an awful lot. I don't think maybe you do like to argue. You certainly have been nice enough with me. Yeah, I don't know you that well. I got it.
That being said, it is a boon to mouse genetics and all other systems we can actually do genetic experiments. We want to modify in certain ways to actually do the experiments. So anything human-wise, though, it's just a lot easier and cheaper to screen embryos.
Well, I'll chime in here because I don't know that I agree. There's scare tactics going on, talking about coming up with modified humans, but it's illegal to modify human embryonic stem cell to pluripotent stem cells.
In the U.S.
What's that?
In the U.S. it is.
It's illegal in most countries in the world. But I don't think that if we're going to go down the road to designer babies like you're suggesting is the hype is telling everyone. Instead, I think that this is going to be good for fixing diseases, genetic diseases. For example, if you have HIV, maybe what you could do is you could get some hematopoietic stem cells from that person, and you can modify it so that the receptor for HIV is missing on the T cells, because there's a natural deletion in the CD4 receptor for HIV, and there was recently in the news there was a stem cell transplant where the person that received the transplant was HIV positive, but received stem cells from a donor that happened to be missing the crucial part of the HIV receptor, and when that new stem cell took over and the immune system was basically replaced with the HIV-proof white blood cells, that person has not had HIV return. You can also, if you have someone with type 2 diabetes, for example, you could come up with, you can make beta cells that aren't going to be recognized by the immune system, and put those into people that have diabetes and can cure their diabetes. So I don't think that it's necessarily going to be germline modification that CRISPR is so important for. I think it's more for fixing genetic diseases. And then
I had that caveat. There's a little one sentence in my chiming in where I had that caveat. But yeah, no, you're right. You're right.
Okay.
Well, and I think those are two good examples. So maybe where we're at in our view of CRISPR is that it is. I mean, it got the Science's Breakthrough of the Year award for 2015 when they released that review of the year, because I think people, as Joel mentioned, they're recognized that there's applications, but as Dave mentioned, things get hyped or kind of, certainly talked up in terms of the broad nature of, like, it's going to do everything. So we're probably somewhere in the middle between kind of what everything you keep hearing about versus what's going to happen as it kind of gets played out in the future. But those examples that Joel brought up, those are really interesting examples for the types of applications that I imagine these groups are thinking about.
The HIV one has the backstory for the receptor is awesome. There's it's like Algerian prostitutes that didn't get HIV and then they realized they had this deletion or something, and then yeah, it's an awesome backstory.
Wow, I didn't know any of that. So my immunology knowledge is not up with that. But yeah, so we're not going to. The other reason that CRISPR has been in the news a bit, which I don't think we're going to touch on as much, is because there's currently kind of a bit of a battle being fought about who to provide credit to for the major discoveries.
This paper is actually directly relevant to that.
Is it? Okay. Well, so there is some of that going on right now, where there was a I think you would call it an essay or maybe a perspective piece by Eric Lander that was published. I believe it was actually titled "The Heroes of CRISPR," and there is a lot of debate about. I'm being polite, so Mark's going to start laughing at me down there in the panel. There's a lot of debate about how he presented the history of CRISPR. And there's some suggestion that he's presenting it in this way because there's patent battles to be fought. So certainly with a new technology like this, we're kind of starting to see like the legal, ethical, societal side of things get wrapped into the science, which happens a lot with big discoveries.
Laura, did you get a chance to see what I had happen in my class?
No, I didn't. What happened in your class?
I like to do one-word responses.
Okay.
I had my 49 or 48 freshmen, and I say the word bacteria, and then I collect one-word responses, and I did a number of things. And as a lark, I did CRISPR, and one person wrote chutzpah, and I have sent that to everybody because that is just fabulous, and they didn't know why I was laughing, and of course because of this. Yes.
Yeah, I think. I don't know the history and the backstory and the contribution of each group in detail, but there are certainly a number of groups that have contributed to advancing CRISPR as a field. And I think we're going to kind of continue to see some discussion happen about, what the roles of those different groups were. This paper comes out of a group out of Massachusetts. So this is a particular one of the groups involved with the not only with CRISPR but with the discussions that are happening now about who do you credit, who gets the patent, so forth and so on. But this paper actually I really enjoyed reading it because I'm used to hearing about Cas9, but this paper actually talks about an entirely different endonuclease and in an entirely different CRISPR system.
And that is the rub to the whole patent disagreement too.
Oh, why is that? Why is that the rub?
The way patent law is written, it's the CRISPR-Cas9 patents are broad, but the way it's written, if you can have an independent system that's not related, that works differently, you can get a different patent for it.
I see.
So this paper actually describes exactly that, where there's a different system, where there's a fundamental mechanism difference between them. And it's potentially easier. That's why without going too much into if you read the discussion, it's very much written contrasting this system with the other system, partly for that reason, to kind of play up the intellectual differences between the two.
Well, and yeah, I agree with you. I mean, that was, as I was making notes going through it, they were very clear on their points about what the sequence and structural differences between the two types of systems are. And they are many, so I don't think they're stretching to call it a different class of system. It seems to operate fairly differently.
Yeah.
Is this the group that had the fast track patent granted, though? Okay.
Yes.
That's what
Currently, they are considered the patent holders, and the Doudna group is challenging that or appealing it.
As my understanding is they had filed a patent, but they did not use the fast track.
Oh, maybe that's it.
So these guys, so Zhang was the fast track, and their patent was for germline editing in cells, in human cells and mammalian cells. Doudna's was just for editing. So Mike Eisen had a good post where it was the metaphor he uses was if Kary Mullis, when he invented PCR, if he just patented PCR, and then Zhang came along later and patented PCR for human cells, that's kind of the
I see.
parallel.
Okay.
So it's a discussion about, so Zhang got the patent first for germline editing human cells and fast-tracked. But in Doudna's patent, which was, I think, filed, so the paper came out first. Her patent was later, but the extension of so her patent is only for the nuclease activity, not for editing explicitly. And so it's basically implied that it would be useful for editing down the road, and that's what the whole intellectual disagreement is about now. The implicit versus kind of explicit things said in the patents. Plus Zhang's was awarded first and fast-tracked. So.
Got it. So, I mean, this to me is like fodder for patent law classes.
Yeah.
many, many years from now. Right now, and then I think this will probably be a great case study. Okay. So that gives us some perspective on why CRISPR is in the news now. But then I thought, let's get into the paper, because I think I really enjoyed this paper. I thought it was very well written, and I have to say I really liked the figures. So I don't know. For me, I thought this was a good paper to read. They kind of laid everything out really clearly, I thought. So I think one of the things that we should start with, just in case anybody watches this who doesn't know CRISPR as well, is to kind of set up the fact that what we're looking at here is a system that uses sequences from invading DNA elements. So this can be plasmids, this can be viruses that invade bacteria, and these sequences are called protospacers. And what happens is they get incorporated into an array within the actual host genome, and that array kind of provides an adaptive immune system. And the system combines a nuclease with an RNA guide to try to recognize when that invading DNA is present again, so you can digest it and basically destroy it, prevent it from invading the cell. If anybody wants to add anything to that, feel free.
The paper that first described this was in 2007, and it was in a journal club where I learned about this. But what I thought was interesting from that paper is that the research was actually done at Dannon, which is a dairy company. So if you have an industry built on bacteria as your production line, then phages could be a big problem if they wipe out your culture. And phages are hard to get rid of and that sort of thing. So to understand how the bacteria can become immune to different phages was a worthwhile goal that the yogurt company funded the research.
It was a really smart thing to fund. I used to work in a biotech company, and we would make bacterial polysaccharides from very, very large fermenters. We always had trouble with phage. So this makes perfect sense.
Yeah, I'm glad you mentioned the backstory there, and I think it's just a great example of, you could use this as an example of why you should tell people about, basic science, for instance, because, I mean, this came out of an application in the industry, but, whoever noticed this was like, hey, that's kind of interesting, and then you've got this amazing system now.
Well, they didn't notice at first. It was basically they had a resistant strain of Lactococcus, and then they sequenced it and saw that that was the change. It was in these CRISPR regions, and so that's what kind of spurred them. They didn't know, like, going into it, they had no clue what the change was, and so they just sequenced. They selected for it, sequenced it, and then kind of fit the pieces together with their model in the end.
Yeah. Yes, that's very cool.
So in grad school, I studied rotavirus, and I studied it for the purpose of understanding how it interferes with innate immunity of the cell. So when this paper came around in 2007, I was super interested, but I never thought of any biotech application of CRISPR for editing genomes. But I guess I should have been thinking more. Then Doudna and Zhang would be out of it, and I'd be rich.
It kind of builds on a long history of, so it started with the zinc finger nucleases, and then kind of talons came in. So the short of it is that there was this cottage industry already of, if you can cut the genome in certain ways, you can get modifications to happen. And it was all about which technology you could use to direct the nucleases to the right place. And so there was zinc fingers, which kind of bound in a loose way. Talons was a little bit more programmable, and then CRISPR's ends up being, you can actually target it to exact nucleotide regions. So that was I think that a lot of people are making the case too now, within the whole patent war, that really there was this other lineage of, you needed to understand that nucleases could modify the genome, and then CRISPR was a natural extension of that too. But yeah, so it's also a story of science building on each other from different directions and then coming together. So.
Yeah, which I think is always very important to emphasize. And I always like it when science writers build back to kind of set the foundation for where we are now, because none of this is done by just one group. I mean, everything is really building off other people's work. So I'm glad when that gets featured. Yeah, so this particular paper, the main thing they were doing, and I'll just kind of show, if I can, figure one here, because what they've done is they've got a contrast between what we're more familiar with, which is Cas9 up here, and then this Cpf1, which is what they're focusing on. So there's a couple of differences that they point out in the paper between these two systems. And the major purpose of this paper was because, as I understand it from their introduction, before they did this work, people had identified the Cpf1 system, but they weren't sure if that Cpf1 protein actually acted like an endonuclease, if it was part of a functional CRISPR-Cas system. So it had all the bits and pieces that they expected, with the exception of a couple. And what they wanted to know was, does it actually target and cut DNA? And so that's what they spent a lot of time in the paper looking at. All right, so I think if we want to get into it, I think one of the first things they did was they started to look at just kind of trying to demonstrate that, yes, in fact, that this system has all of its important parts. And so one of the things that I thought was interesting was, one of the very first things they did was they looked for the motif that is an important part of actually recognizing the target sequence. And that's the PAM, which, for whatever reason, it is escaping me what PAM stands for at the moment. Protospacer adjacent motif. Thank you. And that part, I thought that was pretty interesting, because I was not really familiar with the structure of the CRISPR system, and so the idea that there's this motif that needs to be there, which is part of why, as I understand it, part of why the CRISPR system doesn't target the actual sequence on the bacterial genome. So you've basically taken this target sequence, you've got it in the bacterial genome, but you want to make sure that that target sequence doesn't itself get targeted by the CRISPR system. So the only way that CRISPR is going to target that sequence is if there's this very short stretch of a sequence motif that's upstream of the target sequence, which should only happen in the invading virus or the invading plasmid. So
Correct.
part of what they wanted to do was identify what that motif was. Yeah, please.
I'm just going to mention an experiment that I screwed up. I was trying to get CRISPR to work in some HEC293 cells. It's a cell that you can easily just transfect with plasmids. They take them up. So I threw in the Cas9 protein, and then I threw in a second plasmid that had the guide sequence that I wanted to be made. And I wasn't thinking, and at the time that I designed the guide sequence, I had included the PAM sequence right next to it, and so what I think I had trouble seeing activity from that initial experiment, and in hindsight I was creating a target. The plasmid would be targeted by the Cas9 itself and cause the plasmid to be degraded. So if you're going to make a guide RNA plasmid, don't include the PAM.
Well, but that is a perfect illustration of how they figure out the PAM for this new system. That is the experiment that they use, just in reverse.
Right.
Yeah, exactly. That's true. Yeah, so that's basically what they did, was they created a plasmid library with a lot of different short stretches of sequence to try to figure out what is the active PAM sequence. And so they've got this little figure here where they look at the PAM sequence, and pretty much what it boils down to is it sounds like you really need 2 T's, and then it's kind of whatever is fine. That was what I got from it.
I think it ends up you really need the middle T. It helps to have another T there, but then the third position's kind of fine, whatever.
Yep, and that's what I was going to say, is that their data shows that the second T, so this T here, is really the key one, and then you get a little bit of wobble with the first T as well. So again, I have to say again, I really liked their figures. I thought this figure, it was very clear what they wanted to say. It was like, yep, all right, sequence motifs. I like those weighted sequence motifs. So they've got the PAM identified, and they're starting to kind of demonstrate that the endonuclease itself, the Cpf1 protein, is cutting this DNA. So then they go on to try to look at what do you actually need in order to get this system to work. This is kind of wherein they emphasize, as Dave mentioned, they spend some time emphasizing the contrasts with the Cas9 system. And one of the things they mentioned is the lack of something called the tracer RNA. I like how I'm doing like my poor man's like screen share here. But this is the tracer RNA, which is present in Cas9, and you can see that we don't have an equivalent, a homologous region in Cpf1. So they kind of wondered, like, is it there? Does it not need it? And as it looks like from the data that they show in Figure 2, from what I can tell, they've concluded that the Cpf1 gene is necessary, the array itself with all the spacers that can be recognized is necessary, and then that seems to be it. Because they still see cutting activity, they still see cleavage activity in the absence of any of the other, Cas genes, in the absence of any other kind of RNA element. They seem to think that that's all that's required is the protein itself and then the array. Did anybody else have any thoughts for that?
Well, just to bring it back to the patent war, the ongoing patent war. So just to highlight what Doudna actually did. So originally, you need Cas9, you need the tracer, and then you need the guide RNA. So Doudna's group, in their paper, they were the first to publish that you could design it so that the tracer and the guide RNA were one piece. And so they basically made a 3-component system to 2 components, and then that was their, that was how they got it to work in the cells. So again, this parallels what the paper's kind of doing here as well to make an independent point, but.
And the argument here is that I think from what I'm reading from the discussion is they make the argument that this system might be simpler to work with. Do you think that that's the case?
And other people can definitely chime in. I haven't worked with CRISPRs at all. I just kind of read about it. But when you need one transcript with the tracer RNA and the guide RNA, it's long, like 100 nucleotides or so. And so in order to actually get cells to transfect with it, you need this 100 nucleotide piece, which is kind of hard to synthesize and make. Whereas with the Cpf1, you might only need 30, 40 nucleotide piece, which is much easier to synthesize, and potentially has less of just less challenging.
Right. And I used an in vitro transcription kit that was just a T7 promoter, just a T7 in vitro transcription kit, and I made gobs of RNA, and it was pretty easy to get just piles and piles of the RNA, and so sure 40 nucleotides is probably going to be more efficient, but the 100 nucleotides I made buckets of it, and so that's a little bit of a weak argument, I thought. But the other thing that they're trying to point out with this figure is that there's all different sorts of CRISPR-Cas systems, and they're into 5 types, type 1, type 2, all the way to type 5. And then within these types of CRISPR-Cas systems, there are different classes. The class 1 CRISPR systems, they require a protein complex, so multiple proteins, for the endonuclease to work properly. Whereas the Cas9 that we usually talk about, it's from Streptococcus pyogenes, and it's from a class 2, and because it's class 2, it only needs the 1 protein. And so, the Doudna group or whoever showed that you could have just 1 RNA and connect the guide to the tracer RNA, and then this shows that you just need the 1 RNA. So it's just going to show that you only need 2 components, 1 protein and 1 RNA, to get the job done.
Okay. Yeah.
So it also ends up kind of looking forward, and they don't go over this, but if you read some of George Church's stuff, what they're trying to do, when you're trying to manipulate multiple parts of the genome too, and you have to put in multiple guide RNAs, the smaller they are, the easier it is to target different regions with the same. And so, yeah, you can get a bunch even if it's 100 nucleotides, but it's thinking downstream applications. Shorter is always easier for things, I think. But.
Got it. Yeah. Okay. That's a good perspective to have on that. Before we move on from this figure, I just have a general question. So the top of this figure shows they've got the spacers and then the direct repeats in between the spacers. And then they kind of looked at small RNA transcription. So they're looking at levels of the different spacers as they're being transcribed. And I was just kind of curious, like, why is this one so highly, how does the transcription of each of these, because it's under 1 promoter, right?
Yeah, it's 1 transcript.
So why are we getting such a high proportion of reads here? I couldn't really think of an explanation for why that is. Does anybody?
That would suggest that that particular RNA species either has a longer half-life, or an alternative idea would be that it doesn't efficiently have a target to recognize. I used to study microRNAs, and microRNAs, if there was a lot of target for a specific microRNA, you could end up getting the microRNA to be degraded, and that was the idea behind these things called microRNA sponges, is you'd put a transcript into the cell that had multiple copies of the target for the microRNA, and the microRNA would be used to target that sponge, but in the process the microRNA would get used up as well. So it could be that if it's similar to that system, maybe the target for that CRISPR guide isn't around. But something to do with stability would be my guess.
Okay, that makes sense.
It's not really likely, but it's also possible that there's a region of the genome that kind of almost matches it. And so it's not gonna, it might be just an alignment error thing, but I'm not sure about Francisella. So.
Okay. Do you think it would? Okay, so if you had like a prophage or a plasmid that got integrated into the chromosome before it could have been digested, would that?
Yeah, if there's another RNA that's like 1 base pair off or 2 base pairs off, and you're just looking at all the reads and aligning them to this target, then
That could be too, yeah. Okay. Yeah, it just stuck out to me. I was like, what's it do? What is that? All right. Okay. Yeah, so then after they've kind of demonstrated what the components are that are necessary for activity, for endonuclease activity, and seeing the cleavage, then they kind of go on to look at the particulars of the what they're calling the CR-RNA. So the RNA that actually goes and targets and binds to the protospacer sequence in your invading phage or your invading plasmid. And so then they kind of talked through what's necessary for the activity. Magnesium is necessary, so they see that they need magnesium, they need Cpf1, they need the guide RNA. But then the thing that they point out here, which they also, again, make as a contrast with Cas9, is that the resulting cut is actually a staggered cut rather than a blunt cut. And so this is, they make an argument that makes the process of using this for editing simpler, because rather than relying on homologous recombination, you can now actually have a piece that takes advantage of the staggered cut in order to put it in there. So that is a little bit of what they look at there. And then also they'd start mentioning here, I think this is where they talk about the fact, or maybe I'm skipping ahead of myself. This kind of gets into figure 4, where they start talking about the nickase versus having a complete double-strand break. And why are they making the comparison to nickase? Why?
So the thing here is that Cas9 cuts bluntly and it cuts both strands. So in order to work in human cells or anything, what CRISPR-Cas9 is actually doing is you're targeting the region of the genome to modify. It's cutting both strands, and the non-homologous end joining fills in a slightly error-prone way, and that's where some of the mutations come from. In order to get Cas9 to not do that, there's some mutations that have been found where it turns Cas9 into a nickase instead of a nuclease, which then allows you to, instead of cutting both ends, you can actually put in other pieces to recombine in, not through non-homologous end joining, but just through basic recombination.
Okay, got it. All right.
And so if this is a nickase, then it just implies that it would be, you wouldn't necessarily need to rely on non-homologous end joining, which a lot of bacteria don't have any. So this is one of the reasons why you can't do CRISPR-Cas in a lot of bacteria, just for genome editing, is because they don't have non-homologous end joining. So kind of one of those funny. Sometimes it's easier in human cells to engineer things than bacteria. But.
Okay, got it. All right. So what they find here is that it sounds to me like that is not there are no mutations that turn Cpf1 into a nickase. You either get the double cut or you don't.
Yeah.
That's my understanding.
Thing. Yeah. It seems like a restriction enzyme kind of a thing, right? Where you just have an overhang and.
That's what I thought. Yeah.
And then
I think they make the suggestion that it might have to act as a dimer.
They do, you're right.
So if it's a dimer, then your nuclease domain from each of your dimers cuts at a slightly different spot.
Okay.
And the other point that they make that's relevant too is that with Cas9, when you cut the genome and modify it, you basically are modifying the target site, right? In some way, whereas this one, the Cpf1 seems to cut away from the site, and so you can therefore use the same target site again and again and again.
Yeah, you're right. I remember them mentioning that. So it cuts away from the target sequence, which is good. Yeah. Okay. Yep. So they talk about that. So they talk about, and then they look at the catalytic residues. So it looks like as predicted, the catalytic residues that they targeted are the important ones. And then they looked into kind of observing the nicks. It doesn't show nickase activity. So then at that point, they start getting into what are the requirements for the CRISPR RNA in order to see this nuclease activity in vitro. and they end up doing a number of different alterations to the sequence. So they look at length, but then they also look at the importance of particular base pairs, and then they look at the stem loop structure. So these are all features of the guide RNA. I think I'm using the term guide RNA correctly there. And trying to figure out what's necessary for that, for the activity of the system to still work. Like what alterations to the guide RNA can be tolerated and you still see cleavage activity.
Yeah. So in my YouTube channel, these little diagrams. I have dry erase boards. And so this is basically the guide RNA. The red part is the guide RNA, so that's the part that's going to base pair with the genome. There's one nucleotide here that tends to be a U, and then it forms kind of a stem loop, and then it has some extra sequence here. So for 5A, they truncate this part back a ways to see how far they could go. For 5B, they make mutations, and basically using similar nomenclature to the microRNA literature, they call it the seed region, and they found that the seed region is up close here to the U. So the part that's important for recognition is up near here. They tried in 5C, they chopped back on this part of the CRISPR RNA, and then in 5D they did mismatches here. So any mismatch messed up the activity. If it was only 4 that had base pairing instead of 5, that was a problem. And then for 5E, they tried swapping out that U and trying different things. They also had some different sequences right here on the stem part, but it was this U that was important. So those are the features you need. You need, out of 24 base pairs, I think you only need like 16 was the minimum amount. Out of these double-strand the base pairing, you need all 5. You need the U. I can't remember how far back you could chew on this part before it lost activity. But anyway,
yeah.
I just thought that might be a good way to go over that.
Yeah, that's fabulous. Thank you. I think that was really well illustrated. Yeah, and so I was curious about you. On your whiteboard, you had pointed out for 5B, they were looking at what you were calling the seed region, and they were looking. That was when they just changed the base pair or changed the nucleotide. Sorry, for that area that actually matches the part of the guide RNA that's actually complementary. So at that point, is it the position, or is it actually a nucleotide change? Like, is it the nucleotide that's important, or is it the position important, or is it both that's important, or?
I think that they'd have to go and do an extensive, more extensive experiments where they came up with new guides and new targets and doing it. But my hunch would be that just like in microRNAs, base pairs 2 through 8 of your microRNA are known as your seed region, and mismatches there are far more important than mismatches elsewhere. Mm-hmm. Here, all the mismatches right at this end seem to completely get rid of the activity, and all the mismatches down here, it acted fine. And if it was just like a base pair mismatch, or the base that they chose to substitute in, I think that you wouldn't have such a nice clear cluster right in this area. It would be hit or miss all the way down if that was true.
Yeah, that makes sense. Yep.
That is a good figure.
That is. Thank you for making that. That was very helpful.
It's a really useful resource. I'm serious.
Yeah, it is.
What I hope is that showing the YouTube channel will inspire some of my students to do the same thing for concepts in my classes, and I think that'll help them and help others, so bravo.
Yeah, no, that's true. It's really helpful if you can break a figure down that way for when you're reading for classes. For any students who are watching, there you go, do that.
Well, not to keep plugging my YouTube channel, but I use those for my videos on YouTube, and it helps me keep going forward with my discussion, but it also it reminds me what I'm talking about and whatnot. But I don't know. Go ahead and take a look at some of those videos and see what you think.
Yeah.
As they say, like and share.
Yeah, there you go. Okay, so then they've kind of done a lot to characterize this Cpf1 system. They've made some contrasts with what we know about Cas9. But so far, if I understand correctly, they've basically been working in Francisella. And I'm so sorry that Karen is not here since we're talking about Francisella. But what they then wanted to do was try to figure out, does this...
They're working in E. coli with some Francisella stuff in it. Basically.
Do what?
They were just working in E. coli with some Francisella stuff in it. So.
Oh, that's true. That's true. But Francisella gets represented, so...
No rabbits died in the making of this experiment.
Yeah, no tularemia was caused, especially by a Novicidae.
Yeah.
Okay, so then what they're going to do is they're going to look at Cpf1 family protein diversity. So now the question is, we've characterized this Cpf1 system from Francisella. So now does it occur in other bacteria, and what is the diversity of it? And so again, I gotta say, I just really like their figures. So they did a whole, I mean, you can't read this. Go look at the paper. But basically, they found it in a number of different systems. So they found it in a total, I think, of 16 different bacteria. And when they were looking at just the sequence, they were looking at, I think this is for 6B, they're looking at the direct repeats. And they essentially found that there were 3 species that looks like they were divergent. So we got these 3 here.
And so one interesting thing to point out, just from an evolutionary point of view, some of these bacteria have both the Cas9 system and a Cpf1 system. So what that means is completely up in the air. It's just an interesting little, something's going on there where they're not necessarily acting in the same way, or if they are, it's. I think there's an interesting story behind that. But.
Yeah, I since you brought that up, I kind of wondered about that myself. Just really quickly, I'm getting a little bit of an echo off of somebody. Does somebody have their? Oh, maybe it was Joel. Sorry, Joel.
Is it back?
So what are they have? So if they have 2 systems, are those 2 systems? I mean, if you could look at, I don't know if anybody knows this. If you've got a bacterial genome that has these 2 systems in there, are they targeting this? Are they working independently of each other? Are they working it all together? Do you end up with the same protospacers targeted by both systems? I'm kind of interested in why you'd have both in the same genome.
I doubt you would end up with the same protospacers targeted by both, just because of the nature of how because they're different repeat regions. So they're gonna be incorporated differently, and the repeats in between the spacers are different. But yeah, I don't know why it would. I mean, unless there's, like I said, I think there's probably some unexplored non-redundancy between the 2, that one maybe works in one way and one maybe works in, maybe one is for these bacteria against phage and maybe against plasmid, one might target RNA, one might not, like it's,
Well, Dave, these ones that possess 2, what's their normal environment?
I don't know.
Is it lacno one of them?
Is what?
Lacnos, I can never say it correctly. Lacnospiraceae?
Lacnospiraceae. I can't tell from the figure which ones have it.
Well, I guess what I'm trying to say is, in some ways, it reminds me of having multiple classes of restriction enzymes in the same bacterium. So it wouldn't necessarily be a surprise if they're living in a very competitive environment, say the gut, or the mouth, with a large, for lack of a better term, diversity, microbial diversity there. Maybe they need it all there and don't in other environments. I don't know. You're the evolutionist.
Well, in these CRISPR systems, the open reading frames, as well as the CRISPR spacer repeat, and if you have a tracer, they're all together. And so you can imagine those being transferred en bloc from one bacteria to another. If they were scattered all over the place, it would be very odd. But since it's all together, you can see maybe horizontal.
Mm-hmm.
That's true. Yeah, that's a good point.
Cool.
I will say, so just another complete tangent here, but Mark likes it. So your Pseudomonas syringae video with the freezing of the water. So when I was doing the syringae genomes, and when the CRISPR finder first came out, I threw all my genomes for Pseudomonas syringae into the CRISPR finder, and they all had crazy repeat regions. And I'm like, wow, this is awesome. There's something weird here. And it turns out the ice nucleation protein is repetitive in just a way to be picked up by the...
Yeah, I thought so, because it just yeah. Cool.
So it was like it was excitement about CRISPR, and then like, oh wait, no, that's not.
Well, it was like, oh, the stupid ice nucleation protein.
Stupid? They're fabulous.
All right. So basically, what they found with this is that it looks like the sequence recognition for these three species that had a different sequence. They tried to put it with the Cpf1 from Francisella, and it basically didn't cut. So for these systems, it looks like we're seeing some divergence in the system in terms of sequence conservation and then sequence recognition. So if I understand it correctly, what they were basically saying is that when the sequence becomes divergent enough, you can't interchange that guide RNA with the Francisella Cpf1 that they were originally working with. So there's you need some degree of match between the protein that's doing the nuclease activity and the guide RNA so that there's a recognition there.
I think it was another interesting point that this just brought up too is just so if you just look across the genomes, even with the Cas9 system, there's only a subset of the systems that work, and so really it's this exploring the evolutionary space where you find the ones that work in the way you want them to. So it's just an interesting, we have the tendency to be like, oh, that's present there, so obviously it's working how we think it is, but yeah, it's only a subset that work in this case and the Cas9 case, which is interesting.
Yeah, and I mean, when I was reading this, that was one of the things when they started talking about the evolutionary divergence of these systems. That's something that I mean, hopefully there's somebody working on that, because I think that in and of itself is a really interesting question. What the selective pressures are for these systems being present in bacterial genomes and evolving in these different ways. That in itself is, I think, just from a theoretical standpoint, a really interesting question. So then, of course, because of course, you have to with a CRISPR system, talk about mediates robust genome editing in human cell lines. You got to talk about it. You got to have your happy figure about that. So they did. They tested 8 Cpf1 family proteins, and what I think, if I remember correctly, what they basically found is that only 2 were very robust, and I think one was maybe kind of meh. So what goes into this? Why is it that they've got 8 potential candidates and a goodly portion of them just fizzle?
I think it's proteins. You can build the best construct ever, and then it'll work in a weird way when you actually put it into cells, right? So I think sometimes they're all optimized for different places and in different regions, and not everything works in vitro. So I think that's my take on it, but.
Yeah, they did assays with each of these to see if they would work in just in vitro, and they all seemed to, if I remember right. But yeah, once it got into the human cell, there must be some sort of interference or incompatibility somehow.
It makes you wonder what's, and I'm sure they're looking into this, what the particular characteristics are of the 2 that worked that make them that much better or more efficient than the ones that didn't. But yeah, but then they show that in human cell lines that you can see some evidence for nuclease activity, and so that is part of what feeds into their kind of conclusions and their discussions when they make an argument for this system for all the reasons that we've mentioned. The fact that it doesn't need a tracer RNA, the fact that it's a single endonuclease, the Cpf1 is active. If you have the CRISPR array, it seems like those are the only 2 things that are needed. It makes those staggered cuts instead of the blunt cuts. They kind of wrap all this up in their discussion, and as Dave had mentioned, it's written very much from a perspective of contrasting it with what we know about the existing Cas9 studied system. And so there might be a range of motivations for why they have chosen to frame it that way. But certainly they make some arguments about why this could be a potentially more efficient tool for doing genome editing than what's being used right now, which is primarily Cas9.
So I just tried to skim through the paper to find where they talked about maybe the size of the protein.
Mm-hmm.
I think it was 120 kilodaltons, but I'm not sure. But the reason the size might be important is that the strep pyogenes Cas9 is 160 kilodaltons. This Zhang group also found a Cas9 from Staph aureus that was something like 100 to 120 kilodaltons. And that's a big deal because a lot of times if you're wanting to use CRISPR for changing things in human cells, you might want to use a lentivirus to as your vector for getting that into your target cell. And lentiviruses have a limit of how much sequence you can put in, and the strep pyogenes Cas9 at 160 kilodaltons is a pretty big sequence, and so you're almost maxed out on what you can put into the cells. Whereas the Staph aureus and I think this one are both enough shorter that there's plenty of room to do some other stuff with that leftover space.
So this one is the monomer is 150 and it elutes at 300. So that's page 762, bottom right. But yeah, so it's like 150, I guess. But.
Okay, so maybe Staph aureus is better as far as that that idea of being shorter. That was from the same group as Feng Zhang's group, and that was a Nature paper, I think. But they basically put it through the same steps as here. So there's like, you find something you think is a Cas9-like enzyme, and you run it through these exact same steps. And so, while this paper isn't super interesting as far as uncovering a new way things work, I think this paper is really informative as far as what steps you would take one after another to characterize a system like this. And so I think that right now I don't have a class where I have undergrads read primary literature, but this might be one worth running through over the course of a few days to break it down. They first look at the protein, you next look at the Cas9, next you look at where is it in the other bacteria genomes, do they all work, and finally can you get it to work in human cells. It's just a very, there's these are all obvious things that they should do when they saw that the CPF might have Cas9-like activity, but they just do it very nicely.
Yeah, I agree with that. I think, as I've said a couple times, I think they've laid it out really well, and their figures are really well organized. And as Joel showed us, you can break them down really well into kind of a nice summary where each panel is showing you a different piece. So yeah, we are going to talk about CRISPR in my genomics class, but I didn't set aside paper discussion time for CRISPR. So I may talk to them, I may pull some figures from this, but I don't know that we're going to get a chance to read a whole paper about it. Dave, we did read I made them read part of the human genome papers. I did it. But I think they're doing okay with that.
Leviticus for biology, that's all.
And well, hey, I am at a Catholic institution, so they should be ready for that. Yeah, so yeah, this was a great paper, and I really appreciate the suggestion, Joel. So thank you very much. Did anybody have any, before we close out, because it's getting on 4 o'clock, did anybody have any closing thoughts about the paper in general? I'm going to take that as a no. So thanks everybody for being here. I really appreciate it. I'm really glad we got started again for 2016 and we could find a good time for most people to make it. So we'll do another one in February. If anybody has a paper they would like to suggest, absolutely feel free. And that's certainly the people who are here now, but if anybody watches this and they would like to knock off a paper on their to-read list and then discuss it with people, we're always looking for suggestions. We're a very general microbiology journal club group. We've done some stuff with like microbiome things and so forth, but all microbiology is good microbiology, so I'm happy to get any kind of suggestions from anybody. So with that, I think we'll leave it until February, and thanks everybody for being here, and I hope everybody, since it's Monday, I can say I hope everybody has a good week. And we'll see you again soon.
Thank you, Laura.
Thanks for organizing this.